So next up we have Dibyendu. Dibyendu is a postdoc at the Living Systems Institute at the University of Exeter. He works with Wolfram Mobius, and their group combine the physical world with the biological world. They use computer simulations and wet lab biology, and both of those systems feed into one another. Dibyendu did an integrated master's in systems biology at the University of Hyderabad before undertaking his PhD in chemical engineering at the Indian Institute of Technology in Bombay. There he created mathematical models to explain experimental observations of how different bacteria share and exchange metabolites. In his postdoc, he investigates how the structure of bacterial biofilms impact bacteriophages and combines theoretical modeling on the computer with laboratory experiments. And he uses the data from both of those to drive forward one another. Tonight he'll be talking about using these phages, a type of virus that can be used to fight bacterial pathogens. Everyone give Dibyendu a very warm welcome.
Thank you for having me. So, although you have heard about what I'm going to talk about, no, what I work on is a little bit different from what I actually do. But I'm still going to talk about phage therapy because I'm roughly in the same zone as phage therapy. So, let's first talk about why we are talking about this phage therapy. Today, we are in the world of antibiotic resistance, and that's where we are actually looking for things which can help us overcome the threat of antibiotic resistance, and hence phage therapy has come out as one of those candidates who was very promising. So, as an outline of my talk, I'm going to talk about, first give you an introduction to phages, then I'm going to give you a surprising history of the phage therapy, which is, believe it or not, 100 years old now. But we have largely been neglecting it in the West. Why there is a renewed interest in phage therapy today? What are the challenges of phage therapy? And what are the emerging approaches which many people are researching in the lab? So, threat of antibiotic resistance. If you see this plate on the left, what you have here is a lawn of bacteria, and each of these white spots are different antibiotics which are put on the plate. And you see these clearings which shows that the bacteria is unable to grow in that region because the antibiotic is present. However, when this bacteria becomes resistant to the antibiotic, as seen in this plate on the right, you see that either the region of clearance is extremely reduced or it's not present at all. This is essentially showing us the effect of bacteria which has become resistant to antibiotics or antimicrobials, and presents us a world where even if we have a drug, it will not work, and we will have cases where people would be dying from diseases and would have extreme symptoms which we have in our generation not seen for a really long time. It would send us back about 100 years and essentially cause havoc in the way our society works today. So that's where bacteriophages come in, because these seem to be like one of those players which hold extreme potential to solve this. So phages are actually in reality a very specific type of viruses which only work on bacteria. So they infect and replicate within these cells. This image here shows one bacteria, and it is covered with lots of tiny T4 phages. And these phages usually have a head which is made of protein, in which there is the genetic material, usually DNA or RNA, and you also have a tail, which is the part which it uses to find and attach to the host bacteria. They also may or may not have a sheath. So, although these are the components of a phage, there are a large diversity of phages, and it may or may not have different permutations to combinations. So, believe it or not, there are phages which do not have the head and tail, they only have the sheath, and they are called filamentous phages, and still can do their job. So, before I go forward, I'm going to just play a video to give you a better understanding of what happens in the cycle. So here you can see a phage, a standard one, the head, its shaft, and its tail. It's going to float around the liquid and going to find its way to one of its hosts. When it finds its host, the tail fiber is going to attach. It's going to now then send all its genetic material going to send its genetic material into the bacteria. And once that goes in, the genetic material is now going to be transcribed. So the DNA becomes RNA, the RNA becomes protein, and this protein is essentially going to compose different parts of the phage, which are then going to be creating new phages. Then the cell becomes full of these phages, and the cell cannot stay, and the cell explodes, and the bacteria dies. So this is essentially one way the phage eats up or predates on these bacterial cells. That's why we are talking about phages being predators, which we can use to kill these pathogens. So with this life cycle today, it's easy enough to understand why phages may be a good way to try to kill bacteria. But 100 years back, that's not the case. We didn't know what was happening, but people still observed that phages were able to kill bacteria. They thought of it as miracle liquids, which was suddenly popping up and bacteria was dying. So if we walk into that history, we'll see the earliest known report was in 1895, when Ernst Hancke, who was a British biologist, who was in India because of the colonial rule, was looking, heard about the miraculous waters of healing properties of Ganga and Jamuna rivers. So he got intrigued and he took up that water, and he found that the general bacterial load in that waters was much lower compared to the reported numbers from European rivers, which was surprising to him because the river generally looks very muddy. And this is the other Yamuna river, and this is the confluence. So these two rivers are very different. So when he did that, he took this water into the lab and he tried to grow the cholera bacteria in these waters. And what he found is that cholera bacteria did not grow in these waters, which was very surprising to him, but he didn't have any explanation at that point of time. So he found it as an interesting observation. He did it, he kept it, but he couldn't go further at that point of time. A few years later, in England and in Paris, two different scientists were looking at similar kind of things. So in 1915, Frederick Thwart, he was looking at ways to create vaccines in the early age, and he was looking at growing, so from the skin cells of calves, he was trying to create vaccines. But his cultures were always being infected by bacteria, so he wanted to prevent bacteria from growing. So he was looking at the bacteria in his plates, and he saw sometimes randomly there would be these clearings which would come in bacterial plates. So this is the culture of bacteria, and there's a clearing between. Felix Teherel also saw similar things on his bacteria, which was in case of the dysentery causing bacteria. What they did was they pricked a clean pin into this clearing, and they put it onto another area where there was the same bacteria, and they saw another of these clearing form. They were pretty surprised and they didn't know what was going on. So they tested that solution, took the pin out, put it into a solution, and they heated it, and they saw that this stopped happening. So they understood that there must be something maybe living inside it, or maybe something which is biologically active, which is killed by the heat. So they instead tried to do a filtering such that anything which is as large as the bacteria would not be present. When they did that, they still found that the activity was there. So then they believed that there must be something which is there inside, but must be not as large as the bacteria, which is there. At that point of time, I think they had some clues of what viruses were, but the technology was not up to the mark that they could actually visualize what was going on. So this is also where they kind of were stuck at. But Felix Teherel had the wonderful idea that maybe we could use these to actually treat diseases. And at that point of time in Paris, he tried to use it for a few cases of dysentery in some young kids, and he found good success in them. Later on, in 1923, George Eliva, who was from Georgia, came in, who was the head of an institute in Georgia, in Tbilisi, came and visited him, and he was intrigued by this idea, and he asked him to come along with him, and essentially said that he will give him full reins to look at this research. However, the problem happened that soon the World War II started, and Eliva, after the World War actually, he came on the wrong side of Stalin, and then he was murdered. So all the focus of the research kind of went off, the leaders were changed, and phage therapy, although kept on going there, but the connection to the Western world kind of was broken because people moved on to different directions. Also, antimicrobials were also at the same point of time being discovered and became hugely successful. So while Paul Ehrlich was one of the first to create a synthetic antibiotic or antimicrobial, it was in 1928 when Alexander Fleming accidentally discovered penicillin from slime molds. And later on, Howard Florey and Ernst Chain developed the mass production systems, which helped a lot during the World Wars. So by the end of the World Wars, antibiotics had really found their place, and the Western world was sold from it. And from then onwards, capitalism took over mass production. It was an easy solution, you didn't have to worry much about it, and it took over the world. However, today, as we are overusing antibiotics, and we have the problem of resistance coming in, and we don't want to go back to a world where we would not be able to help our loved ones and see them die in front of us. Hence, phage therapy is something we are looking back at. So while we understand phage therapy has been happening for a long time in the Soviet Union, in the West we have largely forgotten them. But today, more recently in the last decade, there has been some cases of compassionate uses of phage therapy, especially in the Western media in the US, which has given the masses an understanding, a rediscovery of the process. And there has already been an understanding of trying to rediscover this and trying to use it. Why is that we are doing this? It's essentially because today we understand how phages work. Today we have an understanding of their life cycle. We have advanced microscopy tools like electron microscopy or fluorescence microscopy to see what's going on. And we also understand what are the mechanisms which allow the phage to interact with the bacteria. We have also some understanding of how phages or bacteria evolve. And with all of these individual components now available to us, we can tune the phages to do our bidding. So while we have been talking about phage therapy in terms of just curing a disease, there also lies the promise of using phages not only to cure disease, but improve our general health. So we all understand about a lot of gut microbiome balances. Probiotic drinks are always telling you about good bacteria and bad bacteria and how you should use their product to increase the amount of good bacteria. However, with phages, you could also do something similar. If you have a mix of different phage cocktail, then it could essentially keep in check these variety of bacteria in your gut and essentially give it more space for them to not run out. Because there is no good bacteria and bad bacteria in our gut. What essentially happens is if any of the bacteria goes too high in number, then you will essentially get a pathogenic reaction. So phages could be a good way where you could non-specifically control their numbers and increase their numbers. One more thing to recognize is that the threat of antibiotic resistance or antimicrobial resistance does not come from the fact that we as humans have overused it in our daily lives. It's actually because we have used antimicrobials in the feeds for plant diseases, for animal feeds. We have dumped tons and tons of these things in the environment, which have led to an artificial selection on these environmentally occurring bacteria. And hence they have started becoming resistant. Most in our general day-to-day life, we will find antibiotic resistant strains in the hospitals, but we are also starting to see them in the feeds, and that's where the problem is. So if you could have phage therapy work wonderfully, then you could essentially use it for food control and food safety. You could put them in harvest, post-harvest, even in industrial sanitations and preservation, and this could essentially cut down on the antibiotics and take care of the rise of these antimicrobial resistant selection pressure. Now, while this is a big and promising wish list, there are challenges for this one. And first problem is essentially that bacteriophages have a very narrow host range. What I mean by that is usually it is one phage species which can only kill one bacterial species. If this bacteria evolves or changes, or if you are looking at a sister bacteria which is very similar but not the same, the phage may not be able to kill this bacteria. And you would have to find a new phage entirely to kill this bacteria. If that is the case, then in order to develop phages into a therapy, you would have to have a large library of phages, and you would have to screen every time whatever is your disease is, find that bacteria, isolate it, then screen it through the library, find what works, and then use it as a therapy for that person. This is going to be time sensitive as well, so maybe if you do it and it takes you 2 months to do this, and by the time maybe your disease has evolved, maybe you're dead, maybe something else has happened. So it's not the most easiest to get this done. There's also the case that matches may not be found. There's also the case that something which works on a plate may not work inside our body. And one more thing which we have not talked about is that phages are not exclusively a killer of bacteria, but they also sometimes just like to coexist with bacteria. So they become lysogenic. That's something which can also be like break our plans of using phage therapy. There's also the case that bacteriophages have limited effectiveness because they cannot often be used for bacterial diseases which are due to biofilms. So biofilms are when bacteria grow in a thick layer of cells, and they have these slimy layers on top called extracellular matrix, which phages cannot get into. We've already talked about resistance coming in, and bacteria changes and the phage cannot impact it. There's also the case that some of these phages sometimes secrete some sort of toxins, and because they are using it to kill the bacteria. But these toxins can have run immune reactions in the bodies, our bodies, if we use phages directly. So in order to use phages, we have to take care of a whole lot of things, develop new processes, purify them in ways such that these things don't happen. So finally, I'm going to come to what are the emerging approaches we are trying to use. So the wish list, quote unquote, of using phage therapy would be if you could take phages and you could control their host range, if you could enhance their activity against biofilms, if you could create a virulent strain where you could prevent them from being temperate, or essentially prevent them from coexisting with the bacteria and always kill the bacteria, or if you could essentially add to the phages genes such that they have additional genes which enhances killing activity, then you would have the perfect phage therapy. And how do you do that? Today, the most promising effect is essentially bioengineering or synthetic biology. So people are trying to look at the phage genome, and so the RBP stands for receptor binding proteins. So the tail fibers of the bacteria can be modified, and you could essentially replace the gene. So if you have two different phages, and each of these are going to act on two different bacteria, if you could essentially chop the tail of this one and put it onto this one, maybe you then have a phage. So maybe this is a temperate one which likes to coexist with bacteria. If you take this part which identifies the bacteria onto this part, then this bacteria would be creating a new designer phage which can kill this bacteria. People are also trying to add multiple different polyvalency. So what it would mean is that different kind of tails together in the same species, such that the same phage may be able to kill multiple different bacteria. And these are some things which people are exploring, but it's not the most easiest at this point of time. The other alternative is to actually increase the killing activity. How you could do it is essentially by adding certain genes which counteract the bacteria's own defense systems. So many of the bacteria actually have an immune system, even being a small cell themselves. They have immune systems called the CRISPR systems. And using CRISPR-Cas-based systems, you could essentially disable the internal firewall which bacteria themselves have, which renders phage activity null, which nullifies phage activity. There's also the possibility of the phage having some sort of biofilm depolymerization, which will allow the phage to remove the biofilms and enter the biofilms. And it's also possible to have additional enzymes on their tails fibers, which will allow the bacteria, phage, to kill the cell wall, destroy the cell wall, and enter the bacteria much more easily. So, coming to this, so while that is about looking at how to increase the phage's effectivity by itself, there also lies the points which I talked about, being everything being labor intensive. and you still have to do the screenings if you have to find the phage libraries. Towards that, I'm going to do a segue to some of my research. What we have been trying to do is create some high throughput assays to screen these, and for that purpose in our lab, usually what you do for a phage screening is you take this plate and you put agar on top of it, and you put a lawn of bacteria on top of it, and then you put some phage on top, and then the phage. If it kills the bacteria, you'll see them as small spots. But if you have to do it by hand for a large number of cases, you have to do multiple cases. We are trying to use an open source pipetting robot called the OpenTrons system and do this at a miniaturized scale so that in one plate, instead of one phage and one bacteria being tested, you could test 96 antigens. So that's what we have been trying to do with these Omni trays. You put agar, you put drops of different bacteria you want, you put the drops of phage on top of this, which you want to test, and then you could either observe them under the microscope, or you could put them on top of a flatbed scanner even, and see these clearances being formed. And that's what we have been trying to do. So, a small video just shows the robot in action. Just takes in the liquids you want and makes these drops. So this is this creation of the host spots. So we are hoping that this will essentially help us just screen things much more faster and move towards phage therapy much faster. Okay. Thank you.
Thank you so much, Dibyendu. So, other than a phage cocktail, what would your cocktail of choice be here at the Bootlegger?
That's usually long and lasting for me.
Very nice. Anyone got any more serious questions?
Hi, so you spoke about how our overuse of antibiotics has sped up the natural selection of bacteria and sped up their evolution of antimicrobial resistance. What's to say that us doing this more and more won't speed up their resistance to phages?
So there is always resistance to phage. So resistance to phage is people generally term phage and bacteria interactions as arms war races.
Yeah.
So anytime you have a phage and a bacteria coexisting, the bacteria tends to win. So if you use a phage and a bacteria together, that's why phage therapy has not been successful in general. If you use it quick enough, it may reduce your symptoms because initially there will be bacteria killed. But some phages will now, some bacteria will now be escaping and continue the disease. The hope is, or at least the way phage therapy has worked till now, is that you give it a high enough dose, you reduce the bacteria to a certain level where the human system can now come in and take care of it. So all the other effort of improving creating designer phages, which have additional payloads to improve its killing activity, improve its recognition, is towards ensuring that may not happen. But also the good part is that if you actually get, there's often cases have been noticed that if a bacteria gets resistant to phages, they may at the same time get more susceptible to antibiotics. So that is something which people are actually trying to do, that hoping that if phages and bacteria can be used together, or at least in some sort of moderation, then you would have a population which, if you have two bullets to the solution. If one doesn't work, the other may work. You just don't want to be stuck in one solution where you just stuck with no options.
Thank you.
Thank you. Do the phages themselves not get targeted by the body's immune system?
So the phages themselves generally don't. There are a few phages which get, as I said, that some phages have these toxic enzymes on their surfaces. They may trigger immune systems, but it's not very common because phages are, like truth be told, phages are all around us. Any water you drink, anything around us, it must. So I think there is this common quote where like our body has like billions of phages. Our body has trillions of phages at the same time. If we have billions of bacteria, we have trillions of phages. It's just that we do not recognize them. They're so tiny. So phages, some phages can, but most phages tend to not. But obviously, if you're going to develop a therapy, you have to go through all the GMP and protocols to clear that. Hopefully, it will not happen.
Any other questions?
Are there like environmental applications for any of this sort of stuff, or is it just a clinical setting that you'd use these in? Is it too dangerous to like use for something like water treatment or something like that?
No, that's definitely. So the case about the food safety this is where I say example. So that was one example. But definitely, if you could use it, if you could understand phages or at least the diversity of what's going on, then you could target them. But at this point of time, if you say that you're going to just use it for, say, water remediation, you would have to know what's there and then you could do something. Otherwise, it will be like shooting in the dark. So unless we get to the stage where we could create these polyvalent phages, one phage which can kill a lot of different bacteria, or just self-evolve and do their job by themselves, then maybe, but we're just not there. Another question?
Yeah, how is it proposed that the phages will be delivered for human therapy?
How is it that we will
Yeah,
be delivering?
like,
Okay,
or
so
like-
I mean this, 100 years back, people were just creating solutions of phages with no bacteria and just orally or topically using them. In many cases that's still done today, but if you have a phage solution, if you treat it in bulk, and if you just keep it like that, it will decay over time because they are living entity, they are proteins essentially. So even if they are stable, they will not be stable for long enough. So if you want to ensure shelf life, people are looking at ways to encapsulate them in some sort of like lipids or some other proteins which are more stable. But it's like depends on what the use case is. So currently it's going to be topical and just take a pill.
Amazing. Thank you so much, Dibyendu.